Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn. Our guest today is Dr. Thomas Seyfried, professor at Boston College and author of the groundbreaking book, Cancer as a Metabolic Disease. Professor Seyfried, welcome.
Oh, it's a real pleasure to be here. Thank you.
So, tell us about your vision of the origin of cancer.
Well, my view is that of Otto Warburg's view, that cancer is a mitochondrial metabolic disease, as he originally described in his work from the early 1900s through the mid part of the century. He claimed that cancer was basically a disease of energy metabolism involving the mitochondria of the cell. There had been a lot of controversy associated with that view, with evidence that might be, on the surface, inconsistent. But we have recently delved into this in much greater depth and found the missing link, so-called, in Otto Warburg's central theory, which is that they, in addition to lactic acid fermentation, using glucose as a substrate, tumor cells also express succinic acid fermentation using glutamine as a substrate, which we refer to as amino acid fermentation inside the mitochondria. And we think that will resolve a lot of the problems associated with the understanding of the origin of cancer.
So, how does Otto Warburg's view on the origin of cancer differ with what the mainstream is in oncology?
Well, the mainstream views cancer as a genetic disease, and this is clearly stated on the website of the National Cancer Institute. It's clearly stated in the majority of textbooks, biology, biochemistry, cell biology, the medical books. Cancer is a genetic disease, basically driven by what we call damage to proto-oncogenes. We refer to these as oncogenes that make tumor cells grow quickly. And we also have defects in these genes that are supposed to slow down cell growth, the tumor suppressor genes. And almost the entire field has focused on precision medicine, personalized therapies that deal with the various types of mutations that might be found in cancer cells, with the understanding that this is primarily a type of genetic disease. That's what we call the dogmatic view of what cancer is. And that essentially started with the work of Theodore Boveri in 1914, when he considered, from work in a sea urchin, that looking at cancer cells with broken chromosomes, he felt that the origin of the disease might be due to some genomic problem in the nucleus, although he apologized profusely for his profound lack of knowledge on anything associated with cancer. Regardless of that and his apologetic comments, the field seemed to embrace a purely speculative idea from someone who knew very little, if anything, about cancer.
So, in your opinion, it is not primarily a genetic disease?
No, it's not a genetic disease at all. It's primarily a mitochondrial metabolic disease, as Otto Warburg originally described. The gene mutations that we see in cancer are largely downstream effects of damage to the respiration. So the mutations are simply an effect that would be expected from a defect in the ability of the cell to generate energy through respiration. So most of what is being studied in the field today are epiphenomena. They're effects, they're not the causes of cancer. And in my view, this is the reason why we have a continued crisis in this disease, with over 1,600 people a day dying in the United States, and in China maybe as much as 8,100 people a day dying. And people are dying all around the world as the result of this fundamental misunderstanding on the origin of the disease.
And what's the evidence that the origin is metabolic and not genetic?
Well, the origin of metabolic is all cancer cells, regardless of the tissue that they arise in, show signs of fermentation. These are clear signatures of the kinds of energy metabolism that goes on in the cell. So most cancers, in fact, they refer to it as the Warburg effect, an effect where cancer cells produce large amounts of lactic acid. Lactic acid is a waste product of a fermentation metabolism. All cells on the planet were fermenters before oxygen came into the atmosphere. So all organisms that existed fermented before oxygen came into the atmosphere. Cancer cells, regardless of whether they come from the brain, the colon, the bladder, they are fermenters. They will ferment either lactic acid or succinic acid using glutamine as a substrate. This is the common phenotype of all cancers, not the gene mutations, because we know every single cell in a tumor has a different genetic composition. No two people have the same composition of genes, and there's no cancer cells where a gene is constantly seen. So this is what would be expected from a downstream epiphenomenon, not the origin of the disease.
But what about all of these genetic markers like BRCA1, BRCA2 that seem to be so predictive of the development of cancer in BRCA1, BRCA2 case in breast cancer in women?
Well, they're not so totally predictive. They have a penetrance of about 50%, which means that 50% of the women that would have that mutation never develop cancer. Cancer develops in the breast of a woman with BRCA1 only if that gene damages the respiration of the cell. And that's where you see BRCA1 expressed in mitochondria causes mitochondrial damage. Now, why some women who have BRCA1, the same mutation that another woman would have to develop a breast cancer, why that woman doesn't develop the cancer? As Otto Warburg said, there are many secondary causes of cancer. There's only one primary cause, and that's damage to the respiration. If BRCA1 damages the respiration in a population of cells in that individual, the likelihood of cancer will be high. If the gene does not damage the respiration, these women have these mutations and never develop breast cancer. And that's true for every single one of the so-called inherited risk factors. We don't call these causes, they're risk factors. A person exposed to chemical carcinogen might get cancer of the breast or some other organ. But not everyone exposed to that chemical will develop a cancer. So the cancer is the chemical carcinogen is considered a risk factor, not a direct cause. The carcinogen will cause cancer only if it damages the respiration in a population of cells. If it doesn't damage the respiration, the risk of cancer was very much lower.
And is there evidence at the cellular level that damage to the respiration is the primary driver of cancer?
We know if we look at all kinds of cancers under the electron microscope, you have to look at these under the electron microscope. You'll see damage to the structure of the very organelle that's supposed to provide respiration. And in biology, one of the most fundamental tenets of the field is that structure determines function. And this goes back to the evolutionary concepts of Darwin and others, that structure determines function. If the structure of the organelle is damaged from electron microscopy examination, it's very likely that that function of that organelle will be damaged. Therefore, it will not be able to respire, and cells that would have those damages would be fermenters, and that's what the massive evidence in the scientific literature has already shown.
And could you speak to the nuclear transfer experiments that helped you come to this view, which you outlined in your book?
Yeah, well, the nuclear transfer experiments were a series of experiments that were done by some of the leading developmental biologists over the last many decades. So many of these folks are what we would call the specific leaders. Dr. Yanish from MIT, we would have Jim Morgan's group from St. Jude. We have a number of different groups that have done these experiments. And their goal was simply to ask whether or not the nucleus of a tumor cell could direct development. And it was interesting that they did these experiments in an unbiased way. They were not challenging the somatic mutation theory of cancer, which is the dogmatic view, nor were they investigating the mitochondrial metabolic theory, according to Otto Warburg. Their goals were simply to ask whether the nucleus of a tumor cell was capable of developing normal development. And what was found is that basically these, when you put the nucleus of the tumor cell into a new cytoplasm containing normal mitochondria, oftentimes the cells were growth regulated, forming tissues and organs consistent with a normal developmental program. No evidence of dysregulated cell growth was seen in any of these experiments. Now, what was interesting, though, depending on the types of mutations or genomic defects that were present in the cancer nucleus, that determined how far in development that organism would go before it aborted. So it makes perfect sense. If you have a nucleus that has broken chromosomes and missing genes and trying to direct development, when that gene that's missing would be needed to move the organism to the next developmental stage is missing, then the whole organism implodes in an aborted way. And this is what was seen over and over again in these kinds of experiments. So the mutations in the cancer nucleus were not driving dysregulated cell growth, which is the definition and signature phenotype of cancer. They were aborting development. But depending on the kinds of mutations, some of these organisms could go through significant periods of development without showing any dysregulated cell growth. So you have to look at all of those experiments together as a group. No one of those experiments by itself would be sufficient to overturn the somatic mutation theory of cancer. But when viewed as a collection of studies, it becomes very hard to believe that cancer could be due to nuclear gene mutations in the mutations in the nuclear genome. And other studies have shown that if cancer mitochondria in an aggressive tumor cell are replaced with normal mitochondria, you return that cell now to a growth-regulated state. And that was done by Dr. Wang and her colleagues at Baylor College of Medicine. So you can either transfer the nucleus or the mitochondria, and you get normal mitochondria suppress growth, abnormal mitochondria will enhance growth. Therefore, the organelle calling the shots in cancer is essentially the mitochondria, not the nucleus.
So when you say somatic theory of cancer, that's the mainstream view that genes cause cancer.
It's somatic mutation theory.
Somatic mutation.
Somatic mutation theory, which was proposed originally by Theodore Boveri in 1914. And then, of course, if you have germline mutations in the genome, like Li-Frau-Meny, as you mentioned, the BRCA1, and there's a number of these rare inherited mutations. Those inherited mutations will cause cancer only if they damage the respiration, because none of them are 100% penetrant. If you have a gene that's 100% penetrant, like in Tay-Sachs disease, we work with a number of different metabolic diseases, the gene mutations are in fact the cause of that. Because when you have the mutation, the penetrance is 100%. In cancer, we have no gene that's known to be 100% penetrant. Therefore, but every cancer has defective respiration. Therefore, the primary cause is defective respiration, whether it occurs from a nuclear gene mutation, a radiation exposure, a hypoxia, a carcinogen, a viral infection, hepatitis C viruses. Viruses enter cells, damage the respiration, and you put the path to cancer. There's two processes that must happen to get cancer. Excuse me. The first is damage to the respiration. The second is the ability of the cell to upregulate fermentation. Without fermentation, you can't get the cell will not become dysregulated. The genes that control fermentation are oncogenes. They're transcription factors that upregulate fermentation. So obviously, when the respiration is damaged, oncogenes turn on, but they're an effect, they're not the cause. They allow the cell to transition into a fermentation metabolism, and that puts the cell on the path to carcinogenesis.
So fermentation is an older way of deriving energy, and it's less efficient. It's not based on oxidative respiration, right?
That's true.
Oxygen.
These are ancient pathways that existed for energy metabolism before oxygen came into the atmosphere.
So when there is damage to the ability of the cell to respirate, it goes back to an earlier way of making energy, and that's the key problem.
The key problem is as long as there are fermentable fuels in the microenvironment that can drive the dysregulated cell growth, you will then progress through the process, the progression of the disease itself. If fermentable fuels become restricted, that cell can no longer divide. It dies. And this is what the phenotype of cells before oxygen came into the atmosphere was. They were characterized as having unbridled proliferation. and Albert Szent-Györgyi had described this in his 1977 paper in PNAS, that before oxygen, all cells on the planet were in this vigorous fermentation metabolism of unbridled proliferation, and they would die as soon as the fermentable fuels were exhausted in the microenvironment, and the cells would die. And that's basically the strategy that we think will be ultimately responsible for the resolution of cancer. You simply deprive them of their fermentable fuels.
Is there further evidence in support of the mitochondrial metabolic theory of cancer over the somatic mutation theory?
Well, I think you have to look at all the evidence, and you have to weigh all the evidence together. The fact that we see damaged mitochondria in electron microscopy tissue sections of the tissue itself. Many scientists are given misinformation or fail to recognize the full extent of what's happening from cells in culture, because a lot of what cancer involves today is growing cells in culture. So you can see they take up oxygen, they give off ATP. Oh, their mitochondria must be normal. They're dysregulated in culture. So you have to go back to really see what's happening. You really need to look at the carcinogenic tissues from patients or animals that have cancer, and then you can make a decision. If the organelle that makes energy from respiration is defective, it's not likely to work effectively, despite what the cell culture stuff may tell you.
Mm-hmm. So you think that going deep into personalized cancer genomics is going to be a dead end?
Yeah, it's a complete dead end. It's a waste of time and massive money. Because what is the purpose? You're looking for mutations in cancer that's supposed to be predictive of a diagnosis or a therapy when they're essentially downstream epiphenomena. So why do that in the first place? And then the other thing is, where do you get the tissue from to make the gene expression profiles? You need needle biopsies. You have to take a tissue from a living person, and you have to analyze the genetic mutations in that tissue to make a prediction on diagnosis or therapy. Now, if the mutations are irrelevant, then putting a patient at risk doing needle biopsy is unnecessary. So this is the thing. And it's been numerous papers shown in the literature that the very aspect of taking a needle biopsy from a patient changes the microenvironment of the tumor, and therefore puts that, if that tumor is on a stage of malignancy, it can be thrown over the edge to form a full malignant cancer. So therefore, the profile that you get is no longer relevant to what's going on in the tissue itself. And if the tumor is completely benign, why are you sticking it in the first place to get some information from a benign tumor? So the whole concept of needle biopsies for cancer, in my mind, makes no sense at all, and puts many people at risk for making their cancer actually more aggressive and metastatic.
And what do you think is the difference between metastatic cancer and non-metastatic cancer?
The ability to invade local tissue and spread through the body is the major concern, and we and others have shown, especially John Pawlik at Yale and Melissa Wong at Oregon Health Center, have shown that these metastatic cancer cells are fusion hybrids between cells of our immune system and proliferative cells that don't have the capacity to metastasize. You have to realize that cells of our immune system, like macrophages, which are a defensive cell in our body, already have the capacity to enter and exit bloodstream, to move through tissues. These are the behaviors of metastatic cancer. And this is a genetic program of a macrophage in our body. The problem is, when these cells become energetically corrupted, they then are dysregulated, and they spread and invade throughout your whole body. And they ferment. They're powerful fermenters. So it's very easy to kill them once you know that they're fermenters. The problem is, this is not generally done.
Mm-hmm. So you believe that the basis of metastatic cancer comes from the same programming as the programming of macrophages?
Yes, immune cells. So the cancer isn't sensitive. The metastatic cancer cell in our body is, in fact, an immune cell. And it's the most powerful immune cell in our body, making it extremely difficult to kill. So they live in hypoxic environments naturally. So the anti-angiogenic therapies, which are designed to take away blood vessels and this oxygen and whatever, they're not going to work, and they haven't worked. So you can almost predict, going through the types of therapies that we use to manage cancer, why most of them don't work. Why we do radiation? We incinerate a particular region, right? Then we put the microenvironment at risk for inflammation, possible return of cancer. If not, we damage tissue significantly. Why are we using toxic chemicals to stop cell proliferation when you just have to pull the plug on their fermentable fuels? I mean, most of what we do in the cancer field makes no sense once you understand the biology of the problem. And this is the great tragedy. This is the responsible for the crisis we have today. The people fundamentally don't understand the biology of the disease. You know, this is hard to, you need to correct that.
And what causes the initial damage to the mitochondria? What stops cells continuing to respirate?
Well, you can have any kind of an insult to a population of cells. A small hypoxic region, an occluded milk duct. Warburg clearly showed that intermittent hypoxia damages respiration. And this can happen from a variety of different reasons. A wound, a blow, a blood vessel that doesn't fully work in a particular region. You have systemic inflammation. So we now know that the obesity crisis is putting many people at risk for type 2 diabetes and cancer. And if you look at C-reactive protein in the blood of these patients, of these individuals, you'll see it often high. What does that mean? It means that their body is in an inflamed condition. Inflammation damages respiration in particular populations of cells in certain tissues. It may vary from one person to the next. So you have inflammation, hypoxia, exposure to chemical carcinogens. This damages respiration. Oncogenic viruses, hepatitis C, papillomaviruses, these all penetrate and damage respiration. And then you have the rare inherited mutations that put people at risk. So any one of these provocative agents has the potential to damage respiration and increase risk for cancer. So cancer can happen from any one of these. This was referred to as the oncogenic paradox, which plagued and perplexed the entire cancer field for decades. Even in Sid Mukherjee's book, The Emperor of All Maladies, on pages 282 and 303, he struggles with this. How is it possible you can link cancer to a common pathophysiological mechanism? And that common pathophysiological mechanism is now recognized as damage to the respiratory capacity of the cell produced by any number of provocative agents, either inherited or from the environment. But all these are considered secondary. The primary origin of the disease first is damage to the respiration, followed by a compensatory fermentation. And that could be the fermentation of glucose or glutamine, amino acid.
So in order to find the cause, we need to see the effect on the mitochondria first.
If you can prevent damage to your mitochondria, the probability of getting cancer, the risk is significantly reduced. That's the bottom line. And I think that when you go back into primitive populations on the planet, Albert Schweitzer going to Africa, looking at native peoples in their natural environment where cancer was very, very rare, not even recognized, or in the Inuits of Canada and Alaska. These people on when the Westerners first went in there was no cancer in these people. You go and you find cancer is very rare in natural populations. Why? Because they're consuming, their environments are the way they evolved. They evolved in these environments. The other thing that's very interesting is humans and primates like gorillas and chimps have a very close genetic composition. But cancer is so much less in chimpanzees than it is in humans. So that yet their genome is almost the same in almost so many respects. Why is that? Because the chimps are mostly eating their natural foods. They're not being subjected. We have deviated from the path of nature. And one of the consequences is a high incidence of cancer and other chronic diseases.
And how do you think diet interacts with cancer?
Well, I think that diets that put us at risk for systemic inflammation can put ourselves at risk for cancer. So I think that there are certain components in super processed foods that are not healthy that could potentially create an inflammation in our bodies, and that contributes to the risk of cancer. All of these are what we call risk factors. There's no one cause that you can say. These are all risk factors. The only real cause is when that risk factor damages the respiration in a cell. So how do you prevent that? Well, you've got to keep your mitochondria healthy, and what's the best fuel for keeping mitochondria healthy is ketone bodies, which you get when you stop eating for several days or a week. Blood sugar goes down, ketones go up. Mitochondria burn ketones and they get extremely healthy only because they're not throwing out reactive oxygen species. So it's a healing process. And that's why they talk about therapeutic fasting, water only, as a healing. People, this goes back to all the religions of the world. People always knew intrinsically that by water only fasting, one would renew themselves. Well, there's a strong biochemistry for the logic of that. The problem is in Western societies, most people don't do that anymore.
And when you speak about water fasting, water-only fasting, what's the time frame you're talking about?
It's hard to know. It depends on the individual, the age of the individual, whether they've practiced this or not practiced this. So that's why we developed the glucose ketone index monitor to allow us to know, because the idea is you're bringing the person into what we call a therapeutic state, right? Therapeutic ketosis is when blood sugar is down and ketones are elevated within normal physiological ranges. So with the glucose ketone index calculator, an individual, whether they have cancer or not cancer, wants to know whether they can get into therapeutic ketosis. They take a drop of blood and they measure the glucose with one strip, put it into the meter, get the glucose, put another strip, ketone strip, into the meter and get it, and you get the ratio in millimolar of glucose to ketones. And that ratio tells us what degree of ketosis, if we're in ketosis, and what degree of therapeutic ketosis we're in. So this is a biomarker to let an individual know if they're in a state of therapeutic ketosis. And that state would then allow mitochondria to become rejuvenated and would significantly reduce cancer. Now, how long it lasts, we don't know. We haven't done a lot of these experiments. In that same state then also puts massive pressure on tumor cells. If tumor cells are already there, then they need glucose, the prime fuel. The glucose goes down. They can't use the ketones because their respiration is damaged, so they become metabolically marginalized. So it's a very interesting approach to both manage the disease and also prevent the disease.
So the theory has a lot of application.
Yes, and not only for cancer, the same problems of inflammation also underlie type 2 diabetes, a lot of cardiovascular disease, as well as what we now think of dementia and some of these other chronic conditions that are afflicting many, many different people throughout the Western societies.
Well, I hope this knowledge becomes integrated into practice very soon.
Well, it's always easy. It's much easier to talk about it than to actually integrate it, because once people realize what they have to do, they say, this is, you do it and tell me what it's like.
Thank you. Thank you for joining us today. We hope you join us next time as we examine and elucidate the frontiers of science and technology with the thinkers creating our tomorrow. Until next time, farewell.